The next talk is from Nicholas Tatonetti and is entitled AI-Driven Biomedical Discoveries Using Observational Data. So yet more AI guys. Over to you.
All right,
I'll
great.
stand up about 5 minutes before the
Okay.
end so that we'll have a little time for questions, and I know we're starting late. Okay.
All right. Well, it was great. I'm actually rereading the Dune trilogy, probably the first time in like 25 years. And I saw something that I'd never really noticed before or even remember, but there's a few throwaway lines, and they're kind of referencing this past when they had to shut off all the thinking machines. And they had gone amok. They had taken over society and then decided to turn them all off, which is something I never really noticed before, but of course now it seems really interesting and prescient. Also great to follow Larry because we're basically going to use those machines and those methods, but we're going to try to use them for good. We're going to try to use them to discover something about drug safety that we didn't have before. And I kind of zhuzhed up this talk a little bit to make it a little bit more about a story. And I'm going to talk to you about a time when someone kind of came into my life and put me on a track, and kind of a very human-human interaction, and that led to ultimately a discovery, which has been pretty interesting and I think impactful for patients, but then also for what it means about how to use data to understand biology. Does anyone recognize this? I love starting with this slide. I know Larry does. A lot of people. Somebody shout it out. Yeah. So this is a scan page out of Darwin's notebook. I would say about 50% of audiences have at least one person that recognizes this, so it's always nice. I love this because he writes up there, "I think," and then he draws a diagram. And of course, you can start to see that he's putting together this relationship between geography and phenotype. And I love to look at it and think about how his observations from the HMS Beagle were the starting point for so much of what we know about biology. And observations are the starting point for all of our discoveries. And this is not just true for Darwin and biology, but this is also true in the field of drug safety. We have these luminaries in drug safety that really kicked off modern pharmacovigilance. Wittekind Lentz and William McBride that kind of co-discovered that thalidomide was a potent teratogen, causing birth defects in the women that were being given it to treat their nausea during pregnancy. Kind of a really tragic outcome. The tools in which we make these observations are changing and advancing. Before Darwin, McBride, and Lentz, they would use their human senses to gather information and make observations about the world and come up with hypotheses. We then started augmenting these abilities with mechanical techniques. We could invent binoculars and microscopes and microphones to see and hear things that we otherwise wouldn't be able to see or hear. We came up with chemical and biological ways to expand our senses beyond just the 5 that we are familiar with, being able to look inside a single cell and measure the variance, measure the abundance of proteins or RNA. And before, when McBride and Darwin and Lentz were all reviewing data, it was kilobytes, bytes worth of data. You can probably still imagine that synthesis of data that Darwin drew on that first slide. It's something you can really wrap your mind around and think about. But today's scientists are dealing with huge amounts of data, terabytes and petabytes worth of data, models with trillions of parameters in them. It's not something that any one human mind can wrap their head around and then come up with good hypotheses for. And we're collecting data in ways that we never really thought about before. We collect data on every patient encounter. We collect all the thoughts we had about a patient and their diagnosis, all the consulting we did, all the labs we order, all the drugs that are prescribed, all of that being recorded electronically, something that we could then potentially use for other purposes. In fact, there's an organization of different academic medical centers and other hospitals that have teamed up to try to put all of their data in the same format. So we're not restricted to just one site's data. We can then do analyses on a global scale. Odyssey, that's how that's pronounced, you just have to go with it. Odyssey is an organization of over 100 different institutions that have done laborious work to get their clinical data all in the same common data model called OMOP. The goal was to get 1 billion patients' records all in the same model so that I can run an analysis in Los Angeles and then I can spread that around the world and run that exact same analysis at hundreds of different sites, learning in these different data sets about how drugs, what effects drugs might cause all around the world. How do we do this type of analysis when the data are so large and so big? I like to think about data mining or finding these associations, training these algorithms to find these associations for me as a way to make the hypothesis generation phase catch up to our ability to generate these data. So I'm going to tell a little story. The first, actually, I'm going to a word of caution. So first, this is an image generated by AI, of course. And it actually replaced an image that I had just stolen from the internet, like any good academic would, and I used to use. But now I use this one, which I actually have the rights to use. So I really like that. Thank you, AI. There's a problem with using all of these observations. And that has to do with confounding biases. And the reason why, so I kind of invite you on this little walk we're going to go on. So imagine we're walking around this beautiful campus, and we notice that this campus is populated by redbirds and yellowbirds. And we want to ask a very simple question. What's the relative proportion of redbirds to yellowbirds on CU's campus? So we grab a notebook, grab a pen, start walking around. And every time we see a redbird, we make a mark on the left. Every time we see a yellowbird, we make a mark on the right. We get back here, we look at our data, and we see that we observed 5 times as many redbirds as yellowbirds on campus. So what does that tell us about the very simple question we wanted to ask, the relative populations of these 2 birds on campus? That's right, nothing. Because now I'm going to tell you something else. Redbirds, they love people. They know when people are walking around campus, they're dropping food, they're leaving stuff everywhere. When they see people walking around, they fly to them. Yellowbirds just want to be left alone. When they see people, they hide behind branches, they flutter and fly away. And so the behaviors of these birds has invalidated your data set for the very simple question you wanted to ask. And this is not just true in our hypothetical walk around campus. This is true every day in the hospital. The behaviors of the doctors, the patients, the providers, the nurses, the administrators, all affect what we can observe in that data and often make it invalid to answer seemingly very simple questions. So I like to think of every drug order as an experiment that we can potentially learn from. And I like to think about areas that not a lot of attention is paid to. So can we use these observational data to study hidden drug-drug interactions? Can we overcome these biases and challenges to make good predictions? Drug-drug interactions are really important. They can account for a large proportion of adverse drug events, and they can happen. Any 2 drugs can cause a potentially dangerous drug-drug interaction. Understanding these DDIs will lead to better outcomes. So we can stop prescribing dangerous drugs together. That will lead to better outcomes. But I also like to think of them as probes into the human body. And we can use the resulting phenotype as a response to the drug as a way to detect what's happening in the body, to connect the molecular world to the clinical world. This is just a graph to show that as people age, it's very common to take multiple drugs at once. Clinical trials are not regulated to study drug-drug interactions directly. There's often big lists that are generated by pharma companies to say, oh, these are potential drug interactions. There's some tests that are done on the pharmacokinetic in labs, but they are not routinely done in patients. And so often we have to wait until these drugs are used in practice in order to identify these potential interactions. Observational studies, even with all of their challenges, remain our only way to systematically evaluate drug-drug interactions. There exist these post-marketing surveillance systems that have been set up for this exact purpose. After a drug is approved, we want to make sure it's safe, and it continues to be safe. And they contain millions of records of patients reporting outcomes, of providers and doctors reporting outcomes, and also lawyers and drug companies reporting outcomes. You can see some really interesting effects when lawyers try to report a lot of outcomes, especially after big news cycles. And in these spontaneous adverse event reporting systems, you can look at patients' times and you can look at a snapshot of their record. And they're maintained by these large governmental and intergovernmental organizations. But drug-drug interactions are not reported. There are very few, there is a way to report them. It's extremely uncommon. So how are we going to discover this effect when they're not directly reported? And this is where we use machine learning. And we base this machine learning algorithm on how doctors diagnose disease. So when a patient comes in to the office, they might have some disease. But you don't see disease. They don't have diabetes written across their forehead. It lies under some level of detection. It's some unmeasured severe effect. But we know that diabetes as a disease elicits certain signs and symptoms. And these signs and symptoms can be evaluated to form a hypothesis about the underlying disease. And so you can't see diabetes, but you can measure blood glucose. And so you can use those in a model to then infer the presence of the disease. We're going to do the same thing, but for adverse drug events. We suppose that a dangerous drug-drug interaction is hidden and not being reported to us directly. But we also suppose that if it's occurring, it's going to have other more commonly reported signs and symptoms. We can use these harbingers. In fact, we can train an algorithm to identify them and then use that model to look for drug interactions. Basically, when drug combinations appear to cause the effects that we know that single drugs cause. So the way we do this is by gathering up thousands and thousands of examples in a strategy that's very similar to what Larry described earlier, and we throw them at the machine learning algorithm, we throw them at this AI, sure, why not, and we let it learn those signs and symptoms. The one that we focused on was prolonged QT interval, or drug-induced long QT syndrome. The reason why is twofold. The first is it's clinically very important. Prolonging the QT interval can lead to a dangerous ventricular tachycardia, and drugs that cause acquired long QT syndrome or can prolong the QT interval will never make it through clinical trials. In fact, they're tested before they enter clinical trials. And if a drug is found to have an effect on these tests, then it's usually abandoned there, unless there's some very extenuating circumstances. We're very good at doing this test in single drugs. Most drugs that you know and are prescribed today are already put through this test, and they don't cause this. But we don't do this for drug interactions. No combinations of drugs are assayed in the same way and evaluated in this way. And so that led to kind of the story part. So I was visited kind of out of the blue by somebody who said they were a reporter. And they emailed me. They said, I've been working on this story for drug interactions, specifically ones that prolong the QT interval. It causes arrhythmia. And I looked out there, and you're the only one I could find doing this type of drug-drug interaction research. Can I come to New York, and can we talk? So this is back when I was in New York. And I said, of course. So he comes in. We have this great chat. His name is Sam Rowe. He says, hi, I'm Sam. I'd love to talk about it. QT intervals are really important. And there's only one drug interaction known that prolongs the QT intervals, quetiapine and methadone. And he's like, I wonder if you could find more. And I was like, yeah, of course. I could just pull it up in this database. And that's basically what I'm doing there. I'm querying the database I created called Two Sides. You can see it in the picture. And then Sam ended up writing a story about it, so I just wanted to include a little quote out of that story, which is nice, and that really kind of encapsulates how we think about using these data to make discoveries. After he left, it was great, and my graduate student is outside my office, like really nervous and anxious, and I was like, Tal, what's going on? And he was like, how'd it go? And I was like, it was fine, it was great, we had a good time. He was like, that's so cool that he came here. And I was like, am I missing something? And Tal was like, he's a multiple Pulitzer Prize winning journalist. And I was like, oh. Got it, okay. But he's fantastic and warm and a great collaborator, and we ended up working together for over a year on this. And so what I did is I trained the algorithm to look for this effect, the one that he was interested in. It found a lot of signs and symptoms that it would look for, and we scanned the entire database of the FDA's adverse event reporting system for any combinations of drugs that cause these effects, that look like they would be prolonging the QT interval. We evaluated hundreds of thousands of combinations, and our top prediction was between an antibiotic and a proton pump inhibitor. This antibiotic, ceftriaxone, is very commonly used in the hospital. It's given intravenously, and lansoprazole is one of the most commonly taken proton pump inhibitors for, it treats GERD, and people take it, it's over the counter. It's available over the counter as Prevacid. You might have seen it. So these are very commonly used drugs. An interaction that prolongs the QT interval would be very serious and concerning. This is the profile that the algorithm found. This is hard to discern by just the naked eye, but easy for the algorithm. And you can kind of pick out, we highlight some points here that may be why the algorithm ranked this so highly. You can do some types of analysis to have it tell you. But we did one more thing too, and we asked the algorithm to predict a negative control. We wanted to take two combinations of drugs through this pipeline to make sure that our algorithm was discerning between drugs that are going to cause prolonged QT and drugs that were not. And so we had it pull out cefuroxime and lansoprazole, another cephalosporin antibiotic, and that same proton pump inhibitor. We then took these forward from this data set into a completely different one, but asked the same question. So we went to the electronic health records. We pulled about 1 million ECGs, and we asked a question, which was, do patients exposed to this combination of drugs have longer QT intervals than patients exposed to just one of the drugs? And what we found is that in patients who took the combination of ceftriaxone and lansoprazole, we saw 10 millisecond longer QTs in those patients. And we saw that there were more patients, a higher proportion of patients had dangerously high QT intervals, above 500 milliseconds. This is an effect that we did not see with the predicted control. But remember the problem with these data sets. They're all observational data sets, and while we do try to avoid confounding bias, it's very difficult to establish causality using an observational analysis. So the next thing we did is we went down the hall and found a collaborator who is an expert in patch clamp electrophysiology, an extremely complex technique, but Rocky Cass, who is our collaborator, had this high throughput machine. It's really cool. You have this little glass dish with microscopic holes in it just big enough for a single cell to get trapped in. You trap that single cell in and you apply a vacuum on the bottom of that dish, and it blows out the membrane, only exposing the bottom of the cell. So you have intracellular material exposed to the bottom of the plate and extracellular material exposed to the top. You can have electrodes on either side, and now you have an automated patch clamp. No more finicking with the microscopes. And so this is us in the lab. That's Rocky on the left. There's Sam again. And we did a test, a prospective experiment now. We've already seen this effect in humans, and now we're just trying to establish that it's real, trying to establish causality. And we did this in a control, obviously. We then exposed these cells to ceftriaxone alone, lansoprazole alone, and then the combination of the two. You get these really beautiful traces out of this experiment. And so you're basically prepping the cell and the channel on the cell, so it's overexpressing an important ion channel called hERG. That's basically the way that all known drugs that cause this effect, they do it via the mechanism by blocking hERG. And then this peak is the hERG activity being measured. And so what we see here is in the control, we see no difference in ceftriaxone alone, we see no difference as we increase the dose of ceftriaxone. In the presence of lansoprazole, however, we see a dose-dependent response, a decrease in the hERG channel activity. And then if we increase the dose of lansoprazole, we see this really dramatic response of the hERG channel to this combination. We, of course, repeat this experiment for our control. We do not see that in the control. And then this was just single cells. So each of these are traces from single cells. We do that over many, many cells, and we can make these averages. And what we see here is this really beautiful dose-dependent response of the hERG channel to the combination of the drugs, not seen alone and not seen in the control. One thing that we can do with this, now that we have a measure of exactly how much they're blocking the hERG channel's activity, we can plug that back into models that would tell us what we'd expect to see in patients using well-established models. And what we'd see at dosages most similar to what we'd have in our EHR, we would expect to see 10 millisecond longer QTs, almost exactly what we saw in the patients that were exposed to both, demonstrating that not only can that observational data be very useful, but it can also be very accurate and precise. This ended up getting written up and in this article called "Dangerous Doses" or the hunt for dangerous doses. It's the only time I've ever been on the front cover of a newspaper. Actually, that's a lie. It's the second time. The first time was in college, where they have a picture of me. Somebody went around. It was like I was just eating lunch in the lunchroom or whatever, cafeteria. And somebody was like, I want to take a picture of you. I'm trying to find someone eating the grossest, messiest sandwich. And I was like, I'm vegetarian. Like, this sandwich is not gross, but it was kind of like spilling all over the place. It was a very good sandwich. But anyway, so that was on the front cover of ASU's newspaper for that. So this is the second time I was on the front cover of the newspaper. And in fact, this series of articles that Sam and his colleagues wrote about this, which included our study, but some others as well, ended up being a finalist for the 2017 Pulitzer Prize. And in fact, over a drink, I'll tell you why it didn't win, despite a unanimous support from the jury. So with that, I'd like to thank you, and I'm happy to take questions and chat.
Okay, so you're really going to tell us over a drink, or are you just going to tell us now?
Do you want me to just tell you now?
Yeah. We're going to drink short. Yeah, we're going to drink soon anyway, so why don't you?
Well, okay. It's actually, I say it's over a drink because it's a better story over a drink. The reason is because of me. So the Pulitzer Prize is awarded by Columbia. Before moving to LA, I was at Columbia for 10 years where I started my lab. And so we did this study at Columbia. And so this got all the way through. It was unanimous. So they rank them. So I learned a lot about how the Pulitzer Prize works. There's like a jury of people for each section. The jury of people rank them, and the entire jury ranked ours first and some other set second. And then basically it goes into a black box, and there's some Pulitzer person who is in charge, and the president of the university gets to weigh in on who wins the Pulitzer. And despite us being ranked first, we ended up not getting it. The other article won the Pulitzer that year. And we're pretty sure it's just because they didn't want to give it to Columbia. They didn't want to seem as though they were biased. So.
Okay, good. Thank you. I have a question about genotypes of the patients. In other words, what are the routes of metabolism of the two drugs that showed the hERG elongation of QT, and was there any genotype effect when you looked at the heterozygotes or the homozygote knockouts or?
Right. Okay. So we didn't look at different genotypes.
Okay.
So I don't know the answer to that. They are not metabolized in the same way. They're not metabolized by any shared CYPs. So that was not expected to be.
Thank you.
Yeah.
Okay. So a question there. You have to wait for the microphone, sir.
I was not totally clear because you said that no drugs prolong the QT syndrome, but actually a lot of drugs do prolong the QT syndrome. So I was wondering if they were both prolonging the QT syndrome and added effect, or it was truly an interaction effect completely unexpected. Yeah. That was my question.
Great question. Right. So I'll clarify. What I said was there are no there's only one known drug-drug interaction that prolongs the QT interval. There's dozens of drugs that are known to prolong the QT interval, and one of our collaborators, Ray Woosley, I think he's still at in Arizona, he maintains a website called Credible Meds and maintains a really nice website of all of them. So we, in fact, use that to train the algorithm. So that's how we train this algorithm to identify that on single drug exposures. We then take it to the drug-drug interaction, drug combination world to scan for combinations of drugs that fit that profile only when used together, and otherwise do not have that profile. And both of them have been tested before. They have no effect on the hERG channel by themselves. And we've replicated that in our study.
Okay.
Thanks.
We may have something online. I don't know, because I wasn't able to get online as usual.
Yeah, thank you. That was great. So my question is maybe related to Emmanuel's, which is your control to ceftriaxone is ceffuroxime, which is another beta-lactam antibiotic, in fact, another cephalosporin.
Right.
And because ceffuroxime does not give you this effect,
right.
It suggests that the combination effect, where you do see a QT prolongation, is due to the biology of these drugs that we don't know. That's often referred to as, off-target effects. Is that how you're thinking about it?
These are definitely off-target effects. I mean, they should not interact with the hERG channel at all. And so it seems like even and we didn't ratchet up the dose. And you can even see that lansoprazole was very difficult to get at high dose. You saw that I cut it off kind of low compared to ceftriaxone. And the reason is because it was difficult to even get it. It kept precipitating out of solution. So I don't know how high you'd have to go. Eventually, there's going to be some mechanical, like sterics that happen, and you could probably block the hERG channel with enough drug.
But I meant that actually even more than just the effect on hERG itself, which is easy to understand.
Yeah.
But you have off-target effects that, you might have an interaction that causes QT prolongation of a type that we wouldn't have anticipated based on the biology that we know and based on the mechanism of action of the drugs that we know.
And and I totally agree. And that's exactly why I love studying drug interactions, because they start to point at connections in biological pathways that we don't otherwise know exist. Yeah.
I've got one question actually about natural products, and natural products meaning things people ingest because the internet says that they're a good thing for this disease. And what they fail to appreciate is that it might considerably affect the metabolism of the proper pharmaceuticals that they're taking.
True.
Have you done any studies to look at those particular interactions? Because the use of natural products is considerable.
Yeah, so the answer is I haven't, but I'm on an advisory board to a grant that's focused on natural product drug interactions. And they've been focused on things like kratom, and so that became kind of a widespread thing. I guess you kind of like smoke it to get high, but it doesn't really work. It just doesn't work. And so like a lot of like party drugs they're focused on. But not to say they are interested in other natural products. They are interested in things like cinnamon. Cinnamon have other ones. Grapefruit is obviously very famous too. These all do have effects on metabolism, and so they are working on enumerating those effects.
That's good. Larry, the other Larry.
So I love your talk. I wonder if you have looked at the worst thing that people take, in my view, which is metformin, which is taken at unbelievable doses. And if you spend 30 seconds looking at its structure, you realize that it has thousands of off-target effects, and it must be the perfect candidate for your work, I wonder.
Metformin comes up in every search. It's actually kind of difficult to study because I can guarantee you, no matter what models we make, metformin comes up. And then it's like, how do I I don't really have a good answer for that because it does have so many effects. And yet, it's like, how do I follow them up? And it's really challenging. So it's a great example of just like a bane of my existence.
Okay, cool. So last one before T.
Oh, just a really quick follow-up. Do you have any discrimination of what's clinically significant or not?
Well, that's why we do the things like we put it back into the models to see. And the other thing is kind of, and just to kind of thank you for doing this, because I get to get a little bit on the soapbox. The cool thing about using patient data and human data and doing discovery in this direction, right? We're going from patients to the lab, not the other way around. And that means we've already focused on things that are clinically significant. We already saw that they had, that a much greater percentage of patients had QT intervals above 500 milliseconds when on this combination of drugs. We already saw that their QTs increased 10 milliseconds. So it doesn't sound like a lot, but for some of them, that increased them into a dangerous level.
If your QT is already at the
Yeah, exactly.
then it's significant.
Yeah, if your QT is already high, then that's gonna be very clinically
So you
significant.
can use those to make a choice on whether for that patient,
And we
that
do.
drug.
That's,
right.
We do. So I showed you ceftriaxone and sorbitol, but we did about 20 different drug combinations. And then in the lab, we've tested this one and one other one. And they both show similar effects. The other one's not as clear. But you can use the data from patients. We have tons of data. We're getting more every day to guide that clinical significance question. And I think that's a really powerful way to use this type of analysis.
Okay, thank you, Nicholas. Thank you, everybody. We'll
Thank
come
you.
back at 1:15. Thank you.